sur
casque.
une caméra ou autre, il faut le mettre en mode casque.
Ok,
Et là, nous, 1 ou 2, on
d'accord.
touche pas.
1 et 2, on est bon.
S'il y a juste une galère,
Pourquoi ça clignote rouge?
parce qu'il n'y a plus de batterie.
Comment ça, il a plus ça? Comment ça, il a
Si on,
plus de
si on
batterie?
descend, c'est catastrophique en termes de qualité.
Oui, parce que là, je vais plus avoir d'image. Il me faut une rallonge pour le brancher.
Rallonge électrique.
Ouais. Hop. Où ça? Ouais, bah ça c'est bon. Il me faut juste le truc de charge et ça devrait aller. Ouais. Un, deux. Ça marche. Donc il y a les deux micros. Donc ouais, il faut mettre celui-là pour l'enregistrement online.
Ok.
Connexion.
Si ça a plus de batterie, le chargeur est là-bas.
Le chargeur. On a des piles de rechange, c'est ça? Non, parce que ça c'est
Surpris, nous, c'est.
ça, c'est
Ouais,
sur
moi j'ai
Là, je passe les doigts. Je suis devant la caméra, normalement, tu dois me voir. Tu me vois? Et est-ce que là, tu as le son? Parce que moi, il enregistre bien, en fait. Tu vas voir ma tête très près, normalement, à un moment. Ce truc, c'est pas possible.
Quel imbécile, j'ai oublié qu'on avait enlevé la prise.
Ah,
Non, de toute façon, je vais le faire à la main.
ah putain.
Je vais le faire à la main, sinon je vais pas y arriver.
J'arrive pas à l'appeler, à chaque fois il essaie de m'appeler. Attends. C'est bon, il a dit.
On l'éteint tout, du coup, on laisse tout comme ça.
Toi, tu prends ça?
Oula, oui,
Tu
d'accord.
t'installes?
Ok.
C'est bon?
Hmm?
C'est bon?
Attends, attends.
Non,
Non. Ouais, 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 ouais. Attends, je suis pas prête du tout, mais c'est pas grave.
Et le clip, c'est bon?
Ça,
Mets-le dans ta
je
poche.
suis pas du tout l'habitude de ça, c'est pas grave.
Bonjour à tous. Ça fait plaisir de vous voir aussi nombreux aujourd'hui pour ces 60 ans, cet anniversaire du LBT. Je suis Guillaume Sterneman, vous avez dû me voir dans les mails qu'on a envoyés, on espère pas trop nombreux pour permettre l'organisation de cette journée. Et je vais tout de suite laisser la parole qu'on a organisé avec Fabio Serponnet qui est ici au premier rang et Chantal Frézot qui va nous donner quelques mots d'introduction pour commencer.
Okay, so welcome everybody. It's very nice to have all of you here. And we are here to celebrate 60 years of Laboratoire de Biochimie Théorique. It was founded in April 1959, so it's exactly the good time. And at the beginning it was a Service de Biochimie Théorique. And it's a nice occasion to come back to everything that happened in the field of Fabio, Marc, and Victoria, who did a lot of work to put this program on. And this program, there will be some historics of the laboratory and the field scientific of molecular modeling and how it is linked to the LBT with Richard and Philippe and Marc. And then we have invited speakers that will contribute to give more importance to this topic. And what I will speak about now is I leave all this historic, all the science to the Richard and Philippe and Marc. And I will speak of people because it's all of you people who were members of the LBT that have made the laboratory and his evolution. And so this is what we found when looking at the documentation on the laboratory. The laboratory had 5 directors: Bernard Poulman, Albert Poulman, Richard Lavry, Philippe de Remaux, and Marc Baden. There has been 50 PhD theses in the lab, and probably more, because the beginning of the lab, people were hired before they had their PhD, so it's difficult to find in the documentation when people had their thesis. Around more than 50 researchers with permanent positions were in the lab for small time or long time, and we had about 30 postdocs and numerous visitors and numerous master students. And what amazed us when we prepared this event was all the different trajectories that people could follow when they left the lab, because the lab was developing the field of molecular modeling, and people who left the lab, they had nice careers, they made research of their own outside the lab, and they spread the molecular modeling domain in several places. So just in few minutes, this is how people moved. So we start from people who moved inside the IBPC. So you have Edith, who changed the lab, and but she remained at the third floor. But now she has a terrace, and she can use sunglasses. But Benoit, he went in the basement, and he cannot use sunglasses, and he works for the whole IBPC. I mentioned Michèle Landez. Not all of you know that she was secretary of the lab before being the secretary of the IBPC. She was secretary here in '61. And then there are people who worked in the lab for some time and then went to make new experience outside and then came back, like Philippe and Charles, Marina more recently. And people also who were in the lab and changed their field and came back to IBPC, because IBPC is a great attractor. So you have Karine, you have Mark Dreyfus, who did his DER here, and Ingrid, who also came back in bioinformatics. And just a few slides to see how the people have spread. So they have spread in Paris, within Paris. They have spread in the Ile-de-France, in the region outside Paris. So people, I forgot, probably I forgot somebody, but, and now some people went all over France, in Strasbourg, Nancy, Rennes, Nantes, Toulouse. Toulouse, it's the old quantum chemist who went to Toulouse, Marseille. And we can continue with Europe. Some people just left for even Finland. And finally, in the whole world, we have here Remo, who comes from California, and who was in the lab at one moment. And some people just made big travel and came back to France. Now we have Karine, who is in Australia. Okay, so it was just a presentation of all of you, and now I will leave Richard, and left Richard, sorry. Je te donne ça. Given by Diane Rousseau, who is the documentalist. When you speak, you have your
it's a pleasure to be back again and see so many faces that I know. It's a doubtful honor, but I spent half of the 60 years of this laboratory here, so it has some history for me. I'd like to start with a little bit of history about the lab before I was here, and I'll get on to a bit of science at the end. So if you've had a chance to walk along Rue Pierre-Marie Curie, you've seen the signs that resume the debut of this building. So it was initially conceived in 1927 and opened in 1930, thanks to finance from the Baron de Rothschild. And the Rothschild family has continued to be associated with the Institute since that time. And it was mainly the result of the efforts of Jean Perrin, who had, with his work on Brownian motion, confirmed what Einstein had thought was the mechanism of that observation, and also used it to calculate Avogadro's number, which was a fundamental step forwards in understanding the atomic and molecular nature of matter. But as far as LBT goes, the Laboratory of Theoretical Biochemistry, the story starts, in fact, just before the Second World War, when Bernard Pullman, at 19 years old, left Poland and came to France to study at the Sorbonne. Unfortunately, it wasn't the right time to come, because a year later the country was invaded and he joined the Free French forces, and that interrupted his scientific career. He became an engineering officer with the Free French and specialized in building and blowing up bridges and demining. And when he was elected to the Academy in France, there were at least 3 generals in the audience, which were his ex-colleagues from the war period, and reminded us of when he used to crawl along the sand of the Libyan desert, probing with his bayonet whether there are any mines in front of him or not. So he met Albert Pullman just before he went off to the army. They opened to women very easily. So as one of his first female students, she was really at the forefront of calculations at that time, and interested from very early on in whether quantum mechanics could be applied to chemical problems. She looked at many aspects of organic chemistry, the nature of the hydrogen bond, conjugation, and how that could be studied with, at the time, very expensive but relatively simplistic quantum mechanical models. Yeah. Okay. After getting her doctorate, she started working in the Institut de Radium, which is just over the way here, in the group of Raymond Odell. And at that time was already starting to look at the problem of the reactivity of polycyclic aromatic hydrocarbons. So PAH are very dangerous compounds, can provoke cancer, but if you look at the family of polycyclic aromatic hydrocarbons, there seemed to be no order in it. Some were extremely dangerous and some seemed to have no effect whatsoever. And one of the very early works, initially by Albert Pullman, later with her husband Bernard, was to show that there was a correlation between the reactivity of certain bonds in these molecules and their carcinogenicity. And that was really a remarkable step for the period. When I was looking up the history of that time, I was really quite surprised to see that Bernard Pullman was invited to lecture at the Chester Beatty Cancer Institute in London. That's now called the Institute of Cancer Research. It's the biggest cancer research centre in the UK. And he was invited in 1953 as a quantum chemist to talk to clinicians about possible links between chemical reactivity and cancer, which I think was quite remarkable for that period. So that was one of the reasons that René Wormser, who was then administrator of the institute here, invited Pullman to come and found the Service of Theoretical Biochemistry. They had published in 1952 this book on the electronic theory of organic chemistry, organic compounds, which had had a significant impact in the field because it was the first time that people tried to put together an explanation, a quantum chemical explanation for a lot of the phenomena involved in organic reactivity. As you can see here, this is actually Madame Berthaud here, who was the specialist of this electromechanical calculator. It's a Frieden. It used to be in the lab when I was here. I don't know if it's disappeared now. It was an expensive machine at the time. I looked up the publicity from the company, Frieden, and it said 10-digit addition, subtraction, multiplication, and division, fully automatic. So all of those early calculations were done on the Frieden or by hand, or done initially by hand and then checked on the Frieden. The Frieden was not an easy machine to use. Did you ever play with it, David? Yeah. It used to get jammed. Yeah. First of all, it made a horrible noise while all the gears were whizzing around, and then from time to time it would get completely locked up, and you needed a specialist to know where to bang it to get it free again. That was the early days of computing here. So the service was formed in 1959, and only 4 years later, Pullman was elected as administrator of the institute. He'd already been named the very first professor of quantum chemistry here in the university, so it was really a period for breakthrough studies at that time. This is the early 60s, early to mid 60s, and at that period, you can see people we know here. That's Bernard Pullman with his wife. That's Pierre Claverie. That's Simon Diner. I think that's Armel Denny. I don't know who that is. Anybody know who that is? Ah, yeah, très bien. And this is Daniel Piazzolla working on the IBM 1130, which was the first machine we used when I came here. So this is the early 60s. At that time, it was very much centered on quantum chemistry or quantum biochemistry. There wasn't any modeling as such. At that period, modeling hadn't really got off the ground because there weren't the computational means to do it. During this time, I'd finished my studies in England, in Manchester, in chemistry, and I went off for a postdoc in Bangalore, where GN Ramachandran had just come back from Chicago and set up a unit of molecular biophysics in the Indian Institute of Science in Bangalore. By that time, he was actually more interested in the four-color problem than in biochemistry, so I spent most of my time talking to one of the postdocs in the lab, Ashok Kolaskar, who is here. Ashok was very, very helpful for me. He not only took me around Bangalore and showed me how things work there, but he also helped a lot getting the computations off the ground, which was not trivial because the only computer was in Madras, which was about a 6-hour train ride away, so it wasn't a trivial start. Ashok became later on the Vice Chancellor of Pune University and then the Science Advisor to the government and the head of biophysics for India. He's had a very illustrious career and also quite a few jobs in the States. But the first time we met the Pullmans was actually here in Bangalore. They were on a lecture tour around India, and GNR asked Kolaskar and I if we wouldn't take them out for a trip. So he hired a car and told us to go to Mysore and show them the palace there. The palaces in Mysore are very famous. They were built by Tipu Sultan. But we never actually got to the palace on the first trip because GNR had organized hotels for the Pullmans, a very good hotel in the center of Mysore, and a very, very bad hotel for myself and Kolaskar near the railway station. But the Pullmans had done their research before they came to India, and they knew that there was an even better hotel outside Mysore on the hill called the Lalitha Mahal, which had been built in 1921 for the visit of the Viceroy of India by the Maharaja. And they said, actually, we'd rather stay there. Unfortunately, that was about 4 times more expensive than the hotel that had been reserved for them. So We made a quick call to GNR and he authorized it, and we were able to drive them up the hill, and they had a very nice stay in this very beautiful marble palace, which is actually modelled on St Paul's Cathedral in London. And they were kind enough to invite us to breakfast, so I've actually been inside the Lethe Mahal thanks to the Pullmans. And because of that, Ashok got invited to Paris, to this institute, for a 3-month stay, and I got offered a postdoc. And that possibility was due to Noad, who just got into CNRS at the time. Yeah? And so his ex-grant was free, and I was offered that. But I also had an offer from Aberystwyth. When I did my PhD in Manchester, I'd been working on the absorption of small molecules on metallic surfaces, and there was a young Cambridge professor who'd just been appointed in Aberystwyth, and he offered me a post, so I had to choose between the two. So when I got back from India, I went to have a look at Aberystwyth, and it looked something like that. And I saw more sheep than I saw people. And I decided that perhaps Paris was a better bet. So I came to Paris about 3 months late, as far as I remember, and Pullman wasn't happy at all, but he kindly kept the post open for me, and what was initially supposed to be a 1-year stay turned out to be 30 years before I moved. So when I came, it was an interesting time. The theories that Albert and Bernard Pullman had been working on to explain the carcinogenicity of polycyclic aromatic hydrocarbons had finally an explanation. Particularly the group of Jarina in the NIH had finally worked out the chemistry of their activation in the body. And it turned out that they went through a complex series of metabolites, the formation of an epoxide, that turning into a diol, a new epoxide, and finally that epoxide breaks and a carbocation attacks DNA. So my first job when I came here was to try and look at how that proximal metabolite might interact with the double helix using quantum chemical calculations. And this setup. So we only had punch cards at that time. I used to show punch cards when I gave my lectures on modeling, but I stopped because none of the students had any idea what it was about. So these cards here were made on this machine by, did the second interpreting step to have the text written on the top here, along there. Cards had a lot of disadvantages, in fact. This is the reader, so you would put your cards in here and they would flash through, except if it was a damp day. In that case, they would get eaten up by the machine, and we had a special knife with a little hook on it to get out the bits of card that got stuck inside. The other thing you shouldn't do is drop them, because if you dropped them, you had a couple of 100 or perhaps 500 or 600 cards that you had to resort into order again. And Christine and I remember very well being just outside here when Amiram Goldbloom came down the stairs and tripped and dropped 2 boxes of cards, and he hadn't interpreted them. Okay, so that was the beginning. And the first paper was on that story, how DNA might interact with this proximal metabolite of BPDE, a particularly dangerous hydrocarbon which you find in cigarette smoke if you smoke. And we sent it off to the International Journal of Quantum Chemistry, and the reviews were quite good, except for the last remark. It came back and said, the science is quite interesting, but really the manuscript needs to be reread by an English speaker. And later on, Christina reminded me, we spoke to Jackie Plumbridge here, who is also English descent, and there was still a major problem because Jarina had showed that not every guanine in DNA was attacked. Very, very few were actually reactive. So we were interested to try and understand what it was that were making certain guanines more susceptible to binding BPDE compared to the other ones. And the first idea we had, because we'd been already, Madame Pullman had been working with Scrocco and Tomasi to calculate electrostatic potentials of macromolecules by adding together the potential contributions of smaller parts, in the case of DNA, bases and sugars and phosphate groups. And so we thought it might be the electrostatic potential distribution around DNA. And that caused immediately a problem of graphics, because at that time we didn't have any means of graphing the distribution of potential on the surface of molecules. There weren't any of the molecular modeling programs that exist today, molecular graphics programs or graphics terminals to do it. So this is one of the very early attempts. You can see it's a very low resolution plot. This comes from one of the machines I most hated in the world. This is the Versatech electrostatic plotter. This was a company that belonged to Xerox. But unfortunately, it wasn't in a woodshed, as it is shown here. It was in the computer center. And as far as I remember, we got back the results once a week. It might have been twice, but I think it was once a week. So you would write your program, you would send it off to the computer center on cards. It would be run there, and you'd get back a roll of paper, which was about this wide, and you'd unroll it, and what you'd see, at least what we saw, was that. And so you think, oh, there's something wrong with the program. I wonder what it might be. So you look at the program very carefully, but you can't test anything. You know? You can only go through it mentally and try. So next week we got back that. And it took a very long time before finally we were getting this sort of thing. And that served us for quite a long while in publications to represent what was going on the surface of DNA, and how changing base pairs could change the potential and the importance of potential in the grooves, and that was why antibiotics bounded the grooves and so on and so forth. In 3D, we really couldn't do anything at all. There was no question of turning a molecule on a screen. In fact, I don't know if it's really true, but Graham Richards, who was in Paris, but in Dordel's group, not here, he told me that he believes he was the first person to make a colored molecular graphic. And he did that on a cathode ray tube by, first of all, designing the part which was in blue and putting a blue filter over the CRT and taking a picture, and then changing it for the yellow bit and putting a yellow filter over it and taking a picture, and then adding the three pictures together. It was a very laborious process. We had another approach to 3D modeling. That was the first attempt. This was with a Portuguese student, Marçal, Oliveira de Neto, and we were working at the time on tRNA. And we had no idea really what was going on. We were trying to look at in the grooves whether you could bind a molecule there or not, and so we decided to build a model. This is 2 sheets of Perspex held apart by Perspex rods, and then we put cork balls here and wool to show the backbone direction. After having convinced Pullman that we needed to buy some Perspex and cork balls and so on, we spent about 2 or 3 weeks putting this together, and then stood back and looked at it, and there was something wrong. I suddenly realized that we'd plotted X and Y on the axes here, X and Y, and Z was the third direction, except we'd made it left-handed. So I had to take it all apart again and rebuild it with the right-handed. Since then, I never ever make a mistake between left and right-handed axis systems. So this was a big breakthrough. This is the arrival of the VAX 780. This was paid for by the Cancer Research Charity, and this was the inaugural day. You can see some people you might know up here. Sylvie, unfortunately, is not here. That's Christina, that's Michel Londes, that's Pierre Claverie in the back. So this was an incredibly expensive machine at the time. And at the time, thanks to some unexpected money, we'd bought a computer at home. It was the TRS Model 3, and it came equipped with 4K of memory. And you really couldn't do anything with 4K. So we decided we want to extend the memory, and we looked at prices, and it turned out it was cheaper to go to England and buy the chips than it was to buy them here. So we made a trip to England, and we bought 8 2K chips to add 16K to the memory of this machine. And as Christina reminded me, we were shaking when we put them into the machine. They were so expensive. And that was our early days of computing. The big jump through in terms of molecular graphics and made a lot of difference to what we could do here. This is the earliest summary I have of the lab. It comes from the early 90s, and by that time the orientation of the lab had changed significantly. There was a lot less quantum chemistry, there was still some. There was theory, following on from what Claverie had done many years before in understanding the basis of force fields in quantum mechanical terms. There was still work going on in various directions, normal mode calculations, Monte Carlo calculations, and then applications to quite a range of biomolecules. At that time, a lot of DNA and quite a few proteins. Philippe is already on the list there. But I'm obviously not going to try and summarize all that. I'm going to go back and tell you just one story of what concerned me in the lab, and that all started in 1979 when Pullman got a preprint of this article by Richard Dickerson, which was the first single crystal structure of DNA that showed the around the mid-80s. I had a very good colleague who arrived for the first time from the Max Delbrück Institute in Berlin-Buch, in what was at that time East Germany, and that was Heinz Sklenner. Heinz was originally a nuclear physicist, and he changed to working on biophysics. And when he came, one of the things he was thinking about doing was trying to model the mechanics and the dynamics of DNA. This was really before any of the standard molecular dynamics programs had got off the ground. I'll just mention here, that's his son Stefan and his wife Gisela. Stefan today looks like this, and he's a thoracic surgeon in a large clinic in Berlin. The reason I mentioned Stefan, we'll see in a little while. He comes back again. So with Heinz, we started working on the idea that we might be able to model DNA structure. This was one of the very first attempts at doing that. And that led to a long collaboration on the fine structure of DNA, and also many trips to East Berlin. When I went there the first time, I bought this city map, and you can see it has an interesting feature that West Berlin doesn't exist. Yeah? And all the streets just stop. It was an interesting time to go through the border at Berlin. One time I went through the border to meet Heinz, and he met me on the other side, and he looked at my passport and said, "You've got the wrong visa." So I said, "Okay, here's my bag, keep my bag, and I will go back." And a West German guard took me through underground tunnels, and I popped up again on the West German side, came back to the East German control, and they said, "Yeah, what do you want?" And I said, "I'm here for a month's stay." And they looked at me, and I didn't have a single bag with me. And it took me, I think, about 5 hours to get through the border a second time with the right visa. So one of the problems we had with Heinz at that time was we were starting to produce structures of DNA, but it was really difficult to understand the details of those structures. There weren't any useful programs at that time to give you any parameters of deformed DNAs. And so we started off trying to design one of those, and that's work that's gone on for many, many years. These are the initial sketches that I made of the various helicoidal parameters that we wanted to measure. And this is what it was transformed into by Monsieur Roger Astic, who was the draftsman for the laboratory. Previous to that, he'd been the computational man who ran the Frieden. But at this time he did all the drawing. As long as he accepted what you were proposing to him, he would do it very nicely, and if he didn't like it, he wouldn't do it at all. And it didn't matter if you were Pullman, hard luck. So this led to making the curves program, which was designed to doesn't depend just on successive base pairs. It depends on longer fragments of the sequence than that, and also that a given sequence doesn't have a given structure. Sometimes it oscillates between several structures, which is quite important to know if you're trying to understand the reactivity of the double helix further down the line. But that's not what I want to end with today. I want to tell you about the last chapter in that story, which is not about DNA itself, but looking about what is surrounding DNA. So if you want to look at water molecules or ions or proteins or drugs around the double helix, then the difficulty you have is that DNA is extremely flexible. These are a few snapshots pulled out of a molecular dynamics simulation, and you can see that the DNA bends very significantly. The base pairs move around, it twists and untwists, it stretches and contracts again. However, if you have available a helical axis which is dynamic and follows the structure of the DNA, instead of just superposing these different structures here, you measure the position of anything surrounding DNA with respect to this helical axis. So you could get yourself a distance along the axis, a distance, radial distance from the axis, and an angle with respect, for example, to the local base pair. So if you apply this method to looking at the atoms of DNA itself, then this very mobile structure becomes this. So it's gone back to being a linear molecule, not bent, not untwisted, because you're looking at from the point of view of an axis which is following all the movements of the molecule. So having done that, we can look not just at the atoms of DNA, but at the atoms of other things around DNA. And we can plot out in any one of these three variables, the radial variable, so that will be a radial distribution with respect to the axis, or an angular distribution around the axis, or a longitudinal direction d along the axis. In 1D, we can also look in 2D, and this gives you some idea of what you've seen effectively untwisted, straightened, not stretched. And in a similar way, if you look at the phosphorus atoms on one chain or on the other chain, they define a radius which is really quite precise. And again, they all lie in the same position because the DNA has effectively been untwisted. So with those references, we can now go and look at some ions. So here, looking down the helical axis, you can see a concentration, in this case, of potassium ions in the major groove and two distributions in the minor groove. You can also look at the unwrapped bean tin here, that's around the DNA, or you can look radially and you see how deep these ion distributions are. But it gets even more interesting if you do it in 3D. And this was the big surprise for us. Previously, when you look at a molecular dynamics simulation, you have the impression that the ion cloud is more or less uniformly distributed around the double helix. But in fact, if you use this approach and plot out ion densities, you start to see some remarkably sharp and remarkably dense concentrations of ions in the grooves of DNA. So these plots here are at 3 molar, although the overall bulk density of potassium chloride in this simulation is 0.15 molar, so about physiological. So the charges on DNA are drawing the ions in, that's not very surprising, but also drawing them into a very specific pattern dependent on the base sequence of the double helix. You can also break things down, not only into minor groove here and major groove, but also to an inner and outer region. You can see here that some ions are actually in the grooves, and a second group of ions form a crown around the double helix. And that's what we did for a full range of sequences. And we don't have to look at the details here. What you're looking at is the population of ions in the major groove or the minor groove inside the phosphate radius or outside the phosphate radius. And the main thing to note is, at least inside the phosphate radius, it's a very specific story. Each sequence of base pairs leads to a very specific ion distribution. For example, here, if we take GC, this is a G pair followed by a C pair, a GC followed by a CG. And what changes here are the flanking base pairs. And those flanking base pairs change the ion density from 0.3 to 0.9 of an ion on average. So this was an interesting story, because we're getting closer now to perhaps understanding what can make things very specific in terms of reactivity, not just electrostatic potential, not even structure, also dynamics and ion distribution. And we found, for example, that some of the oscillations we'd seen before between different conformations, in particular low twist and high twist, were also associated with the absence or the presence of ions in specific positions in the double helices. And we were also able to look at water distribution around DNA. And this is the first time that I could actually see water shells. Hydration shells are very nicely picked out by variations in water density. You can see it in the plane of a base pair here, or you can see it in this cut through at the level of the helical axis, where in some places you can pick out up to 3 shells of well-defined water. And I'll make another example of that, taking us to a slightly more complex system. If you add a protein into the mix, and so we've got a protein DNA complexes, this is the P22 repressor. And if we look at ion distributions here, you see superposed on the sequence dependence of the double helix changes due to the presence of the protein. This is one of those proteins. It's a dimeric protein that doesn't touch the DNA in the central portion of its 20 base pair binding site, but the central base pairs are strongly biased towards adenine and thymine. And in fact, that can be understood in terms of electrostatics when you look at what has happened to the ion distribution, because these large ion clouds here, potassium ion clouds, have been formed exactly on the region of these 4 central base pairs where that AT preference occurs. This is another way to look at that by looking at ion density changes. So the green colors are reductions in density and the red colors are increases in density. And you can see that the protein has had a really dramatic effect on DNA. It's reduced all of this ion density in the outer region on the distal side of the binding site, dramatically increased the density in the region I mentioned before, but also drawn those ions on the opposite side deeper in towards the helix. So there's really a dramatic electrostatic storm going on when a protein comes close to DNA, which again suggests that there's going to be sequence dependence involved in that. And not only the ions are perturbed, the water is perturbed, and in particular, in this case, you can see that the 2 binding regions of the P22 repressor have completely dehydrated DNA in the regions where they bind. So all the water is chased away, but that's not true in the central region where we want to get these potassium ions close into DNA. There's enough water left to keep them at least partially hydrated, so there isn't a large penalty in bringing naked ions into this interface zone. Good. Last steps. So from the beginning, central to this effort with Heinz was the use of the helical axis. That led us to think about the grooves of DNA and how you could actually define those in a mathematical sense. And recently with John Maddox, we've moved on from knowing where the grooves are to being able to measure locally curvature. So we have a radius of curvature locally and also a direction of curvature. And that's particularly interesting if you now want to look at curved DNA, for example, DNA in nucleosomes or DNA in minicircles. Here's one example of a minicircle. And in this plot, the length of these vectors correspond to the radius of curvature, longer vector, smaller radius of curvature. And their direction shows you how the molecule, in which direction the molecule is bent. You can see here that this molecule has 2 strong bending regions in these extremities here. And you can also see perhaps that even in the parts of the molecule in the center, the bending is never in the plane of the curvature, because it's easier to bend DNA in the direction of the grooves. So if you look at it, the bending direction follows a sine wave, basically, above the plane or below the bend of plane, below the plane, in order to facilitate the bending of DNA with the base pair directions. And here's one step further, so we can combine all of that with the ion densities now. And if you look at a DNA where there are strong local bends, as in this case here, you also see that there's a strong change in ion density, and the potassium ions move in to counteract the higher phosphate density associated with the bending. So that's more or less where we are on that story now. Some 30 years after it started, and we're still thinking about it. And to stop, I'd like to say thank you to all of the people who contributed to this and many other stories, and who I had the pleasure to work alongside. And I wanted to come back. He's on the next slide. So these are the people I could find pictures of. I looked for the most embarrassing ones possible. Unfortunately, some people only have recent photographs and some have none at all. So you can have a look at that afterwards. This is what I wanted to mention. Days when he was first at the museum before coming back to the museum, looking at three-stranded DNA, for example, with the physicists David and Vincent on single molecule experiments. And thanks to CICAM, where we first met a lot of the leading lights in the early days of molecular modeling and molecular dynamics. Steve Harvey and Dave Beveridge down here, Peter Coleman. That's Tamar Schlick from the back. That's Tom Cheetham. So those meetings were really very useful in getting us started. And I'd like to end with one other slide. The young people here now have much to an easy life. You know, you've got beautiful computers. You don't have to wait for graphic results to come back for a week. I'd just like to remind you as well that you have a very nice laboratory, and it used to look like this. This was during the work of renovation on the lab, when water was pouring down the walls from the roof, and we had to move the buckets regularly from one place to another to stop it going down to the second floor. When the renovation began, I think Francis Andre will remember, we had a discussion, and Matthias as well. Should we move out or should we stay during the renovations? And I was going to CNRS the next week, and I asked them in CNRS, what do you think? Do you think it wouldn't be better to move out? And they said, you understand, if you move out, you'll never come back. So we stayed. It was wonderful. Thank you very much.
So thanks a lot for this fascinating historical perspectives and also some of your latest work. I'm sure we have some questions in the audience. Comments?
Yeah.
Casey?
Very nice.
There were very interesting things going on. Clavery's work is very famous. They were also doing some very nice work on deconvoluting exponential signals, for example, which had impact in a wide range of fields. So I think it was a slow evolution, that the computing was becoming powerful enough to start looking at larger fragments and not working, as a lot of work had been done in the early days on single bases, for example, with water molecules around them, or small oligopeptides in water. And it was coming to the time when you could start looking at a large macromolecule. So I think it was a slow evolution. It wasn't a choice by any means, certainly not on my part, but it evolved in that way. Yeah, a lot of people left, other new people came, so yeah, it changed. In the beginning, it was really exclusively quantum chemistry. When I came, there wasn't any question of doing anything other than that. Yeah, so it grew slowly. That's it.
Other questions? I have one actually.
Yeah.
So in the last few years there has been a lot of progress made on nucleic acid force fields. And there were a lot of problems in particular because I think, I mean, from what I understood, you have all these residual degrees of freedoms in the backbone. Do you think we need to revisit some results that were obtained back in the day, I mean, 20 years ago? Do you think, I mean, all the concepts you had from this kind of simulation hold, even now that we have, you know, better models to describe them? I mean, what's your...
Sure. Yeah. And we have a specialist sitting in front of me who regularly redoes everything that people did 10 years ago in 2 weeks on their computer system, and improves things. I think now we've reached a pretty good plateau in terms of force fields for DNA. For a long time, DNA was hopelessly unstable. And in fact, it was these early calculations, being the first attempt to look at base sequences, that we pushed beyond the typical 2, 5 nanosecond, 10 nanosecond, up to tens and then hundreds of nanoseconds, and discovered that DNA unwound completely with the force fields of that time. So now we're talking about regularly 500 nanoseconds or microseconds or many microseconds of simulation, and DNA is certainly stable. The comparisons with experiment are now very convincing. There's always more refinement to be done, but I think we've reached a stage now where there aren't any obvious flaws. There's always a question of balance between base stacking, base pairing, which probably has to be looked at again. Perhaps Modesto will say something about it in his talk. But we've certainly undergone a revolution, and part of that was due to trying for the first time to do large-scale simulations. You know, I've said many times the difficulty before is if you studied a sequence, nobody else would touch it. They'd always choose another sequence and another protein so as not to have any overlap, and the result was you couldn't compare anything. So the advantage of doing things in a large group where we all use the same protocol on many different sequences made the flaws much more visible much more quickly, and I think that produced a big leap in terms of quality. But, yeah, nucleic acids are not trivial by any means.
Okay, let's thank Richard again.
Thank you.
So now it's our great pleasure and honor to welcome Professor Modesto Orozco. So while he's setting his computer, let me do a brief introduction. So Professor Orozco is a professor of biochemistry and molecular biology at the University of Barcelona. I think you spent pretty much all your career there, starting from his PhD in 1995. It's impossible to summarize all the great contributions Modesto has made to the field of biomolecular simulations. But he has worked on proteins, on salvation effects. He's probably more famous, I mean, for most of you, I guess, for his great contributions to DNA and nucleic acid simulations. In particular, like Richard said, trying to improve the force fields. And if you follow the field a little bit, in the last years, yeah, there has been a lot of progress, reparametrizing a lot of force field parameters, and things are getting much better than they used to be. So I think it's working now. So yeah, Modesto, it's your turn. Go ahead.
Okay, thank you. So it's a real pleasure and an honor to be here, and thanks Richard for this wonderful, I would say, not only the history of this place, but I would say the history of molecular simulation. So I think I don't need to convince you, that this is a fascinating molecule. Typically when people work with biomolecules, proteins are interesting, RNA is intriguing, and DNA very often is boring, I will try to convince you that it is not. So no, for most people, DNA is the molecule of life. So this thing that was created for life and actually helped to store information. I'm a chemist, so I will try to convince you that that's not absolutely true. So I use this slide that comes from, it came actually for a talk I gave when my kids were very, very young. So this is a kindergarten slide. So in here, what it is put is in a kind of, I think it's a log scale, is the history of the universe. So January the 1st, 0000 is the Big Bang, so boom. And then nothing really exciting happened until March. It was the Milky Way, boring. And then in summer, of course, the sun appears. And with the sun, it comes all the planets around. For us, action happens in September. So it was like the first actually not bacteria, were archaeobacteria. And 1 month later, bacteria decided that was a kind of good idea to segregate the genetic information into the nuclei. And 1 month later, eukarya decided that it was good to join efforts and to put together and create a multicellular organism. And okay, December is the most exciting month. So nothing. Okay, vertebrates appear here. Dinosaurs appear around Christmas time. They disappear in December 30. And mammals appear here one day after Christmas. And for us, you know, all action was in the last day. So Erectus evolved 10 minutes before 11. Humans appeared 6 minutes ago. And we eat Neanderthals 1 minute ago and became the kings of the world. Okay, so if you look and make a zoom in the last minute, you will see all the important moments in the history. This is the first civilization, and here you will think what we consider is the most important points in the history of humanity. And you can ask yourself, okay, when the DNA appears, and you may be tempted to believe that is around here or perhaps here. No, we know that there are short oligos that were actually there before the solar system was set up. And we know this by meteorites originated out of the solar system. So we are talking about a really unique molecule and with an incredible amount, incredible ability to store information and with an extreme resilience. And I like to put this in context. So what you see here, well, there are some debate, but this is probably the oldest document existing, made by man. It's called the Palermo stone. It's of course, in Palermo, and it was written in the 5th dynasty. Remember that the pyramids were in the 4th dynasty, so it's a little bit later than the pyramids, but actually it's a kind of list, if incomplete, a list of the kings from the 1st dynasty. So the document is not in very good shape. You know, you can see has been a little bit shaken, but it's impressive. It's 4,400 years old. So think for a while what happens with Richard's punch card, or what happens with your USB. I mean, mine's every 6 months break. So this piece of stone lasts for more than 4,000 years. But take a look what DNA has done. This is a sequence of an ancient human from Africa. It is 70,000 years old, and the level of error is none. So it's exactly the same message. And we have sequenced the Neanderthal with very small error of 0.1%, something very small, is between 100 and 200 years old. And this is, as far as I know, is the record. It's about 700 years old. Is a horse from the Pleistocene. And we have sequenced with a small error. So the limit is supposed to be 1.3 million years. Compare this ability to store information, this resilience, with the type of way in which we can code documents. And if you combine this with the ability to store information, you can really, or you can not be surprised by the effort made. This is a paper by my friend Iwan Bernie and other colleagues that actually took DNA, designed a code to code information in DNA, synthesized the DNA in the US. Actually, they ship by mail, regular mail. They make a mistake, they send to EBI instead of EMBL Heidelberg, so they have to post again. It arrives to Heidelberg, and then they sequence and they restore the information with less error. The information you can see here, what is there, it was restored with less error than when the data was sent through internet. And think what are the probabilities if instead of using the 4-bean code of the DNA, you use this Hashimoto DNAs where you have 8 letters. In a few grams, you can code all the information generated by humans from the beginning of civilization. So when you think about DNA, don't think just in a biological molecule, think in something really exciting. Think as a kind of treasure. This is our treasure. And for me, really, the most interesting topic of DNA, the most interesting, the real reason why I'm working in DNA, I'm a theoretician, is because of the complexity of the DNA. So when you work with DNA, sometimes you are working and you need to be worried, as Richard showed, on details. You know, 0.5 angstroms it change in a hydrogen bond can change completely the activity of an entire gene. But in many other cases, you are interested in looking at the entire chromatin, and this for humans is 2 meters. So how can you deal as a theoretician when you have to move from the Armstrong to the meter scale? This is something where my group has been has been working for many years now. At the very end, what you need to do is you need to solve a multi-physics problem, and this is what has been our work, trying to join different levels of resolution to the very safe environment of quantum chemistry, where you know that you are accurate, that you are precise, to the very, I would say, floppy bioinformatics world. So this is where we chemists are more confident. This is where biology is. And I will start, and I think this is very appropriate in this place, making this kind of general picture of the type of simulations we can do with DNA, and focus my attention. I used to spend most of my talks in here, but today I'm going to focus in quite a lot in this area here. Is what can you do using quantum chemistry? So there is a tendency to believe that there is no room for quantum chemistry anymore, at least in the field of DNA. I will try to convince you that there are a few cases where you need to go quantum. And this is an old work. It's a couple of years ago. It's the work of Hansel. And it is a very, I think it's a very neat example of why you need to complicate your life with wave functions and so on. It is an enzyme, it's called MOP-M. It's a kind of topoisomerase, but for bacteria. And this is an enzyme that actually relaxes superhelical torsion in the DNA by cutting the DNA. Then it binds the DNA to the protein, and then the other strand relaxes and go back and generates the DNA again. So quite easy. The problem is that to do this, you need to do a phosphotransfer reaction. So you need to transfer the phosphate to another group in the protein, in and out. And any chemist will say that, come on, you need an hydroxyl to accept the phosphate, because then you have hydroxyl reaction will be nice, is the most obvious choice. But nature here makes things complicated. For reasons nobody knows, there is not tyrosine in the binding site, that is the usual suspect. There is not serine, there is not threonine. And the group that solved the structure, Michael Coyle in my institute, actually were crazy trying to find water molecules and any other things. We take the system, Hansel did whatever he could, all the reactions were impossible until he thought in the possibility to have this histidine here acting as an acceptor of the phosphate. When you do that, you can run a KMMM calculation. This is the quantum part. The rest is a classical. And then you do simulations, get free energy surfaces. From this, you can get what is your reaction, what is the reaction coordinate. You can get the delta G's of activation. And in this particular case, we were able to make many predictions. For example, what happened is you put a tyrosine here. Believe or not, the enzyme is inactive. So the enzyme has evolved for millions of years to have their histidine. You put tyrosine, it's not working. We were able to explain the role of this arginine and many other things. But let me show you a more complicated case. This is something completely, I would say, heretic-like. And this is, I think, a nice collaboration with my friend Philip Hollinger in Cambridge. And we were interested in trying to create new proteins. So proteins that would allow us to create new DNA-like molecules. In particular, this is molecules that actually Richard studies this in the past, and many others are called phosphonates. These are like a phosphate, but instead of phosphate, you have this phosphonate here, where this is a neutral moiety. And then you can think, of course, has many advantages in terms of therapy. And it would be great to be able to synthesize this to have an enzyme that synthesizes polyphosphonates, so DNA-like phosphonates. And so the challenge, of course, is how you modify an enzyme. This is the modified enzyme. This is just a model. We don't have the crystal structure. But we took an enzyme that was processing DNA and then played with the residues to make it able to bind a neutral backbone. This is completely esoteric. So a neutral backbone. Can we do this? Can we make the enzymes work? So recognize the phosphonate and actually catalyze it. Remember that the key point to have a phosphotransfer reaction is to be able to stabilize the phosphate group when it migrates from one side to the other. And this is typically because you have magnesiums in the binding site. But here, the migrating group is neutral. So we are changing completely the chemistry. Can we get it? Can we create an enzyme that is working? And making a long story short, the answer is yes. And this is, for example, this is, for example, a simulation from Vito, where you ask yourself, okay, is this structure stable? And this is alone in this simulation. You can see it is very stable. And you can ask another question that probably you don't ask yourself at the beginning. What happened with the chirality? So this enzyme is prepared to have a non-chiral phosphate group. What happened when you put a phosphonate? In this case, is an ethyl. It's going to be chiral specific. And I'm not going to show you the results, but indeed it's going to be chiral specific. You can make the mutation yourself. You have a methyl or a ethyl bond here. These two magnesiums will flow apart, and the enzyme will be completely inactive. And this is actually something that we test experimentally. But once you have this and you realize that everything makes sense, that the enzymes look like reactive, then the beauty of simulation is that you can actually go and see how it works. And it was a big surprise for me. So this is how the reaction works. This is our model. Remember, it's a model, so we don't have a crystal that this hydroxyl will transfer the proton to the PPI and this will exit. So the interesting point here is when you compute the free energy profile. It's incredible because this is almost the barrier of a polymerase working on DNA. So we have created an enzyme that is able to work with a neutral DNA-like molecule with nearly the same efficiency that with normal DNA. And let me show you and give you a little bit more details in this order. This is even more erratic because what we were interested here is trying to get or to understand how a DNA can work as an enzyme. So of course the proteins are enzymes. Everybody knows ribozymes. So RNAs can be also enzymes. But most people ignore, or don't know that actually DNA can be an enzyme. And the question is how these DNA enzymes work. And we took advantage of this very nice paper by Ponce Saavedra et al. That after 6 years they got the crystal structure of the first DNA enzyme. So it's the enzyme that for the experts looks like a pseudoknot type of structure, and what it does is that it takes 2 strands of RNA and join together. This is sequence specific, which is an advantage with respect to normal protein ligase, and at the end of the day you have a single strand of RNA that has been joined together. So when I show the paper, I ask Juan, okay, take a look and see what the hell is the mechanism, because ligases have evolved through millions of years of evolution to have a specific reaction mechanism, how a molecule that is synthetic, that has not been created by rational design, it's a high throughput type derivation, how it works. So then Juan went to the computer, and after a couple of weeks or a month, I don't remember, he came to my office and said, okay, it's not working. This is not catalytically active. So it's impossible to explain the reaction of this DNA enzyme with the structure that is in PDB. Is something wrong in the structure? There is nothing. It is just that they catch a post-catalytic stage. So they catch how the protein, how the DNA looks after the reaction has finished. So what we did then was to actually go back to the structure. We apply a method that we developed time ago. It's called CMIP. It's a kind of Poisson-Boltzmann type calculation. And with this, we titrate the DNA SIME, to find places where actually you can bind magnesium. Why? Because evolution has found magnesium as a key element to catalyze this type of reaction. And we hope that perhaps DNA SIMEs will work with magnesium. But it was a guess. So we found a place here where it was a magnesium binding site. This is actually a place where there is magnesium in the crystal, but we found these two wonderful spots of magnesium. We then take the structure, put magnesium there, did a little bit of equilibration, adjustment of charges, blah, 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 and ran the simulation. And this is boring, but after we have here 300 nanoseconds, but we did half a microsecond. The structure looks perfectly stable. All the interactions were perfect. The alignment of the reactive group was perfect. So it looks like a catalytically active conformation. So it seems that we were on time to do simulations, to do KMMM, but this is painful. So when Juan showed me the results, I was immediately suspicious. So, you know, how do you know this is right? For example, I didn't understand, looking at this structure, this is the hydroxyl, right? You can think in a reaction where this binds to here, and this proton perhaps may migrate to some place. But immediately you ask yourself, why is not catalyzing DNA? So it's an experimental fact. This enzyme doesn't join DNA strands. Why? When actually the 2' hydroxyl is doing nothing. This 2' hydroxyl is not hydrogen bonding to anyone. So why is not catalyzing ligation of DNA? And the answer came from an MD simulation. So we just take this RNA and then modify by a DNA. And so remove the 2' OH. So, and when we remove the 2' OH, what you find is not a surprise for the expert, a change in the pattern of the ribose. Actually, the 3' move away, all the orientation of the reactive orientation changes, and it's impossible to have the reaction. So it's clearly a model, it's just a model. All this is classical, but actually explain a quite surprising fact. Why this DNA SIME is not able to join DNA molecules, on the contrary, joins RNA. Okay, so we were convinced at that time that the structure makes sense, so we ran umbrella sampling, so built a bidimensional reaction, and you can see here the reaction. It's a asynchronous concerted mechanism. There is only one transition state. First, there is the approach of the phosphate, and once the phosphate is approaching, has approached a lot, there is the proton transfer of this hydroxyl, 3' hydroxyl, to this group here. This is the DNA SIME, right? So you can see that it's happy. We characterize this transition state, and then we do the experiments. Experiment is 24.5, you know, nearly exactly the same that we got from our simulation. So this is fantastic news, and we were very happy when we realized this, but there is a bad news here. Is look at that. This is the reaction-free energy. It means that this enzyme will never work. So you have a +12, so you will expect a negative number. So what are we missing? What is the driving force to make this reaction possible? And actually, this is something that was an interesting lesson, because we typically forget that the enzyme needs to be regenerated. And in this particular case, the free energy came from the regeneration of the enzyme. And it was a nice experience. So it was at the end of the reaction, the proton was here. This is the DNA SIME. And actually, this is not the endpoint for the proton. We found that it's very likely that it will migrate through a water molecule to this other phosphate. This is almost barrierless and very similar in energy. Not much interesting, but the really important point is that when you have the proton here, it is also very close, again through a water bridge, to this PPI. And PPI loves to be protonated. So this is a downhill process. Actually, at the end of the day, you have the proton here in the PPI, and this is quite amazing because when you have the PPI protonated, it's a downhill process where the PPI leaves the protein, and actually this is when you have your driving force. And what was surprised, because we were not looking at that, this is a kind of metadynamics type simulation, but you can see that this is a downhill, so you can do however you want. But the interesting point is that actually we got the two magnesiums out of the binding site. So without pretending, we recover the crystal structure. There was nothing wrong with the crystal structure, even if it was not catalytically competent. It was just that they were at the end of the movie when everything has already happened. And this is actually where they got the crystal structure of this. Okay. What is the role of magnesiums? Remember, we invented magnesiums. They were not in the crystals. There was an in silico prediction. But you can see what happens when you remove one magnesium. This is the normal reaction profile. When you remove one magnesium or the other magnesium, the reaction funnel disappears. We predict it should be not computationally active. It will be not reactive. We try then to submit the paper to Nature Catalysis, and the editor was very nice, and they asked in refreshing Bihai Pande, in science as in life is bad to fall in love with models. So it was just a model, come on guy. I mean, catalysis, but this magnesium may be in any part of the DNA site. So why these are ours magnesium? So maybe, okay, so the proof you have is to substitute the oxygen that you suspect is interacting with the magnesium by a sulfur. So to use thioates, phosphotioates. And this is a pain on the neck because you have to substitute stereospecifically this and this other. And this is very, very difficult from experimental point of view. But we did it. And I'm not going to bother you, but this is the kind of reduction in catalysis that you find. In some cases it's very large, in others it's dramatic. So this is completely compatible with the fact that these oxygens were, as we predict, involved in the interaction with these magnesiums. But it may be still a criticism, and the criticism is, come on, you annihilate a reaction, but this can be for any other reason. You need to recover, and there is a way in which you can do it, is by substituting magnesium by manganese. Manganese is softer, and then it can interact with the sulfur. And these are the experiments, so this is not simulation, these are the experiments, and you see this is the control. When you put magnesium, you multiply by 2 the speed, but looks there is a 1 to 2-fold increase in the velocity when you're actually with the phosphotioate derivative. So it's clear that the model was right, and if you want an extra proof that there was some proton involved, we did, of course, kinetic isotopic effect. So at the end of the day, what I was trying to illustrate in this latest example of the use of quantum chemistry, that quantum has some advantage. So it's not linked to a force field, it's not actually, it's just first principle based, and you can use in a topics that are out of the what is normally studied when you work with classical approaches. You can move and study anything for strain and non-canonical that it looks like. We are working on this, but let me now jump to my second level of resolution. That is, when you are interested in things that are not small enough we developed it, was more than 6 years of work. All the data is available, so you can download the original trajectories. Everything is free. You can double check that we didn't do anything strange. We have in the order of, right now, we're 200 terabytes of data that you can actually interrogate and download. The method work has been tested in a variety of systems, and my take-home message, this part of my talk, is that to convince you that MD has now, so for reasons that were related to the triplex war, we were interested in DNA-RNA hybrids. So This is a kind of duplex that happens very often in the cell. It's in the Okazaki fragments and the R loops. It happens during viral infection. It's something that happens continuously, and we haven't paid enough attention to it. We, with Agnes Noy and others, we characterized that this type of systems were asymmetric because, you know, the DNA keeps some memory of the DNA duplex, and the RNA keeps some memory of the RNA duplex. But in this particular case, we were interested in hybrids when it was an imbalance in the purine-pyrimidine rate. So one of the strands was purine-rich, and then the complementary was pyrimidine-rich. So let's say, to simplify, that we have RPU, R pi will be RNA, DPU, D pi will be DNA, and this will be the hybrids. So in green will be those when the DNA is mostly purines. The yellow will be when the purines are in the RNA strand, right? So in principle should be the same. But when you analyze it through all kind of analysis, this is something closer to DNA. And when the purines are in the RNA strand, this is closer to RNA. And when this is the work of Vito, when he showed me the trajectories, it was something that for DNA people, you know, it's a kind of signal of something happens, because comparing one trajectory to the other, it was a significant change in flexibility, and this used to mean that there is some change in stability. So I asked Vito to take a look at stacking, and look here, this is the DPU, so purines in the DNA strand, or purines in the RNA strand, and you can see that there is a very significant difference. So when you have the purines in the RNA strand, this is more stable. And not only is the stacking, but it's also in the backbone. You see that it's better to have the purine in the RNA strand. But this is just a prediction, can be completely wrong. So we then do the experiments, and this is another long period of wet lab, and there are tons of numbers. Just look at these ones in the corner here. This is the DNA, these are the RNA, but the interesting things are here. This is the melting temperature of a hybrid where you have the purine in the RNA strand. And this is exactly the same, but you have switched. And now the purine are in the DNA strand. And you can see, as predicted, that there is a tremendous difference in stability. We predict also, theoretical calculations predict, that a part of it was because the stacking. So when you have a purine in the DNA strand, means I have a pyrimidine in the RNA strand. And when it's RNA, is uridine. When it's DNA, is thymine. So we can make a very simple experiment that is to change from thymidine to uridine, and you see the decay in stability, or do the reverse, change from ribouridine to ribothymine, and you see the increase in stability. However, you never recover the original stability, so you never get this. So it's clear that it was also true, the prediction that the backbone was making a very important effect in stabilizing the hybrids with the RNA-rich impurines. But okay, does it have any importance at all, or this is just a kind of theoretical curiosity? And okay, so if you take a look at this paper we wrote in a few years ago, we get a clear result that actually further experiments confirm that in order to have RNase H active, RNase H is the enzyme that is used in antisense therapy, so it's the enzyme that degrades hybrids. In order to have it active, the hybrid need to be not A-like, so it should be intermediate. If it looks like an A-like, this is not going to be a good substrate. So the prediction was then the RPU-D pi, which are more A-like, remember the previous slide, should be worse substrate. And this is what you find here. When you compare these two oligos, you see that in here, this is the RPU, right? You see that you still have hybrid after 10 minutes. In here, everything has disappeared after 5 minutes. So this is right. It was a first prediction that was correct, a non-obvious prediction. But you can go further. And this is a completely brand new prediction. So if RPU d pi are more stable than the reverse, they should be dpu r pi, then they should be more efficient in CRISPR-Cas9. So in other words, so if you want to tackle a duplex DNA with CRISPR, it is much better idea to tackle the RNA strand, sorry, the pyrimidine strand rather than the purine strand, because your hybrid is going to be more stable, and then Cas9 is going to be directed more precisely. And this is actually what we prove here. These were experiments done by Monse with the help of Roderick Sigo lab, and this is what you find. There is a dramatic difference in, this is 10 to the minus 3, 10 to the minus 4, difference in CRISPR activity, just with the same, everything the same, but just changing the RNA to tackle the pyrimidine or the purine strand. So again, a non-obvious prediction. And another non-obvious prediction or non-obvious explanation. Why do I get a crystal? So when, I don't know if there are crystallographers here, but for a theoretician, crystallization is something like that. So they mix things and they try hundreds or thousands of possible combinations of co-solvents of different conditions, and at the end, bingo, they got a crystal. But why? And this is the work of Antonia, that it was a kind of proof of concept. So we took Druidicus orthodecamur, the structure Richard presented minutes ago, and this is a kind of, you know, the molecule everybody study, and it's a molecule that has been solved by crystal, X-ray crystallography many times. Most are in this symmetry lattice. There are others in this and others in this. So 3 different symmetry space, different lattice. So Antonio decided to study all of them. These are a kind of painful experiments because you are not simulating a single copy of DNA. In this case, for example, you are simulating 80. So you reproduce the cell, the unit cell, then you add periodic boundary conditions, you put water. So I'm talking systems that can be up to 1 million atoms. We talk about 28 different systems. We have an aggregated timing here of instead of milliseconds. All this was possible thanks to Praise that provides the computer resources that were actually needed to do this kind of massive simulation. Just to give you an idea, this is 40 terabytes of data that are available for you. So at the end of the day, boring to that, this is a bidimensional plot where you have the bidimensional twist till the helical parameters which are introduced, and this is through the MD simulation. So this is millisecond scale simulation. The individual helices, and then look at the displacement of the helices in the XY axis plane, that is this movement, and in the XZ and YZ planes that are this type of displacement. And you can see that for this and this helical, for this and this crystal symmetry, there is a displacement. So in other words, the crystal is lost. This is not a stable crystal. So the crystal will disappear because the strands will slide one over the other. But quite surprisingly, everything is perfect here. So why? Then we did something that is typically never done, that is to look at the papers. This is the same in one dimension. We look at the papers and see what is the difference. And this is the kind of receipt. So simulations I showed you before were done with everything normal you put in a solution in simulation. So we had sodium chloride, potassium chloride. We also had magnesium, but we didn't add spermine. So this is why, our guess, but this is why these two simulations explode when this was completely stable. So we need to prove it. And to prove it is very simple. So this is the control simulation that was stable in water. And this is what happened when this was no spermine, and then you start adding a spermine. So what is the concentration of spermine in the crystal? No idea, because it's not seen. So in the crystal structure, there is no spermine density. So I asked the crystallographers in the institute what is the reasonable choice, and then we take a look at different potential concentrations of spermine. And you can see as you titrate actually spermine that seems to be a kind of co-solute that is put there, nobody knows why. Is actually that is not, you don't see in the crystal because it's not fixed. It's moving all around. It's displaced. You can find a few plots, but in general you don't find a place where there is a neat density of spermine. So, I think I have convinced you that we can study small pieces of DNA, we can study long segments of DNA, but can we really go to the genome scale? Can we really analyze a very, very huge system? Can we go to this level? I'm not going to talk about the coarse grain because there are much better groups here on coarse graining. I'm going to focus at the top level, at the mesoscopic level. Our mesoscopic model is quite simple, and this is mostly the work of Jürgen in the lab, and it was a kind of heritage of the ABC consortium work. And so the idea is that you run a long enough number of MD simulations, different sequences, microsecond scale of this, then you project the covariance in the helical space, and from this you build a kind of covariance matrix that when you invert, they will give you a kind of a stiffness parameters. In principle, with this you can simulate the flexibility. What we realize, however, is that the elastic model that assumes that the molecule is a kind of normal mode-like. So it assumes that the molecule has one minima and it vibrates around this minima. In the DNA, is not exactly right. So this is a kind of study of ABC stuff, and what you have here are different tetramers, and this is a result of some machine learning clustering approach where we define how many clusters, how many states the DNA is sampling. And this is a standard approach that is only one, so it's fully harmonic. There are very few cases where this happens. In average, you have in the order of 4 minima, so 4 states. So it means that in reality, you need to be worried about harmonic deformation around 4 different states. Fortunately, you can use, this is just for 2, but you can use EVP theory that has been reformulated by many people. These are Gerhard Hammer reformulation. In reality, it's very simple. It's just 2 harmonic potentials and a switch is there. When you have here, you have a Hamiltonian that you can use to run Monte Carlo simulations and actually sample. But the flexibility of DNA for a given sequence. But we really want to go beyond, and we want to reproduce Cartesian coordinates. So we want to reproduce also the backbone. And here we took advantage also some output of the ABC work that actually led us to characterize correlations between the backbone conformation and the helical parameters. For example, we know that when the shift is going, to very positive values, you will have a lot of B1, but when it's negative, you will have a lot of B2. So in your Monte Carlo simulation, you can actually generate a random distribution of states, and at the end, they will reproduce the preferences for a given, the statistical preference for a given steps. You can do this for epsilon zeta, but you can do for Baccarin, you can do for many other things. And at the end of the day, what you are able to do is to reproduce a Cartesian-like sampling. And this is a little bit technical, but this is the calibration point. This is a 600 duplex. I encourage you to try to simulate this to atomistic MD. It's nearly impossible. We did this, we simulated this one, but you can see that very quickly, the structure is converged in terms of Monte Carlo moves. This is a comparison. For one case, this is a 55 mer actually. This is a comparison between Monte Carlo and MD, and these different helical parameters. This is the V1, V2. These are the groups, and what you have here, I don't know, red maybe MD, black maybe Monte Carlo. I don't really remember. These are the dot product, essential dynamics. You can see is everything in the diagonal, so it means that simulation is able to capture the sequence specificity and actually reproduce also very nicely then the essential deformation modes, and you can say, okay, don't worry about ND, what about experiment? And this is a comparison. This is a Monte Carlo experiment, and what you find here in red, I think, are the experiments, the Redeman structures, and you can see that the accuracy is extremely large. Even when you look at the very small details, so this is a kind of multimodality in different tetramers. In one hand you have the MD, in the other you have the Monte Carlo. I don't know what is what, because it's nearly impossible to distinguish. And even if you go at the RMSD, this is RMSD for the atoms, so it's not helical anymore. This is atomistic, and you can see that the agreement is very good. What is the efficiency? We were able to test and to check efficiencies between 1,000 and 10,000. It's probably more, but there is a point where you cannot run longer, and these simulations you need to guess, because you cannot take 1,000 base pair DNA and do simulation. It would be impossible. For longer, it would be longer DNAs would be much more than 10,000 times faster. With this, what can you do? Go to the microscope, can study fibers, and this is again the work of Jürgen. And the only thing that you need to do is to put nucleosomes there. So we put a nucleosome that for us is a particle that is put here, is frozen, and then the flexibility goes through the linkers. And how do you introduce the nucleosome contacts? Well, we actually calibrate this. These are the histones for us. We calibrate it by fitting to hydrodynamic properties. In this case, were segmentation coefficients. Okay, what can we do with this? So we now have a method that is derived from first principles from quantum to classical fossil, from classical ND to mesoscopic model. The advantage is that we can simulate thousands and thousands of base pairs and many nucleosomes. What can we do with this? I mean, we can actually try to predict the position of nucleosomes, but this is already known. So this is a very easy experiment we do in the lab. This is the work of Evgenio and Oscar. And this is a type of experiment that you degrade the DNA, you cut the linkers, and then you sequence what is here. And this is the type of plot you find. So it's very easy. That here is a region without nucleosomes, and there are nucleosomes there. To get from here to here is not trivial, but we develop methods to do it. And this is the type of plot that you find. So it's very easy to be convinced that here there is a nucleosome, but really the nucleosome is oscillating. It's not in this place. This is an artifact of the way in which we process. We and others process the data. But what is really annoying is when you have this kind of situation, because it suggests that here you have a nucleosome, and here you have another nucleosome. And then what Jürgen did was to actually move from 1D to 3D. So try to build the structure of the nucleosome fiber with these positions that were experimentally detected, and it's a complete chaos. There are collapses, crashes, it's a disaster, so you cannot really make anything sensible. So what is happening? What is happening is actually this data comes from a pool of cells. What you are really getting is not the structure of nucleosome in one cell, but a kind of average of structures of many, many cells. And perhaps the average picture you get doesn't correspond to any single cell. So we need to deconvolute the signal, and this is what I'm not going to go to many details, but the idea is that we are going to try to deconvolute this experimental signal in a kind of populations of individual cell legs, so individual strings of the experimental plot. What can we do with this? We can grow. So we can actually try, we have this is in the order of 5,000 bases. We can actually join with another 5,000 with a linker in the middle, where the conformation here and the conformation here need to be consistent, and then we can grow up and grow up. And of course, at the very end, you need to add extra data. For example, what Jürgen and Diane are doing now is to add micro-C data. So the idea is that you have family 1, and for Monte Carlo you have several structures that you can sample, family 2, blah, blah, blah. You all have all these families. And for all these, you can compute what is the micro-C map, and you can compare this micro-C map with the experimental one. So micro-C is like Hi-C, but detect direct nucleosome contacts. So at the end of the day, what you are able is to deconvolute this minimum number of, the resolution is lower, it is typically 1 to 5 kilobases, but actually is a type of data that is very useful to have a general profile. So in Hi-C map, what you have is this type of information when you know that this piece of DNA here is close to this other piece of DNA here. And these are actually tackling a kind of loop. And this can be transformed and mined in a kind of time average restraints that we implement in a replica exchange simulation. Is this enough? Probably is not. Because again, Hi-C is an average property. It's not a microscopic property. So we are implementing. This is ultra-resolution microscopy. This is a storm data, and this is a technique that is called oligopainting. I don't know if you look this and this. This is a single cell and give you information of GIS chromatin. What you have here is the centromeres. And this is something a little bit more aggressive. These are results from, the experimental results came from Fraser, and these came from a single cell. And what you have here would be the equivalent to a transition of protein, and this is the transition of a chromosome in mouse between time. So this is what probably chromatin is doing across time, moving from different conformations that are compatible with the function of the cell. Okay, I have one example. I don't know if I have time or it's... Okay, then I will stop here because this is much more than 2 minutes. It's just an example of how can you really push the technique and give something that really impacts experimentalists. And let me just finish thanking the people in the group that has been involved in all these projects. I think I have mentioned most of them. I forgot perhaps to mention the experimentalists working in the M&A SPAR, that are Rafa, Nuria, and Isabel. These are the people in the lab that do the experiments. I think I have mentioned all the others. So thanks for the work and thanks a lot for your attention. Thanks. Do we have questions? So in the last part, yeah, so in the last part, the type of thing that you have is are information, for example, on what is the chromosome reorganization when you actually have, in this particular case that I want to show you was methylation driven. So you can actually see what happens when you activate methylation in the chromatin structure. And something that is very, very, very clear is how genes are activated and disactivated, and this is coupled with the structure of the chromatin. The same we have done for several cases. For example, in oxidative stress, you have the same type of. But in here, the prediction is you need to be careful because you fit the method with a lot of experimental data. So, of course, we cannot fault based on first principle chromatin. We need to integrate in these cases, for example, where Hi-C combined it with MNA-seq and the Tejon mesons. This is a case where we study the impact of methylation, and when you look at the entire this chromosome, this genome, nothing happens. But when you look at the individual, you see, for example, that these are always up and always down here. So this is a phenotype that you can see very easily that is quite curious, the same phenotype of senescence is something that we found quite robust because it's found for all chromosomes everywhere. You see that the blue is methylated, and for example, red is unmethylated. So you have changes in the cis to trans contact, and you have changes in accessibility. But again, I mean, you need to fit the simulation tool with experimental data. We cannot do first principles yet.
In the case of the DNA, RNA hybrid, the experiments which show clearly the role of the thymine method. Do you understand what the method is actually doing?
This is a boring stacking type thing. So it's there is hydrophobic, of course, but you do in the gas phase, you still see this, and these results were supported by couple cluster simulations. So there is a significant difference that are related to the methyl interaction. You have many, and this is what is experimentally agreed when you do this ribouridine into ribothymine or the reverse. So it's not, I would say, you never thought about, because it looks like thymine and the urine are so similar, but it really makes a difference in terms of.
I was wondering whether you're able to reproduce high-C data, which you're not able to into the model. Another question is, since you're forced to rigidify the proteins, the nucleosomes, why aim for this level of resolution? And what's the longest stretch that you can simulate?
Yeah, I mean, we are doing this for the entire yeast genome. So we grow up, so the micro-C data alone doesn't allow you to reproduce the high-C data. So micro-C is 100 base pair, so it's quite local. But actually, when you put micro-C data, for us is the kind of cherry picking. So we start with the 1D. So we build the models, and then we got many. So we don't bias through micro-C data. So we compute micro-C data, and then synthetic micro-C data, and then we combine those families that actually fit the best micro-C data. So it's not we don't impose, it's like a cherry pick, and it's information bias. You will never get from micro-C data this long loop type of things. You, in some organisms, for example in Drosophila, you get most of the high-C contacts just by making chromatin restraints, when you put the colors of chromatin. So you put that the polycomb are close to the polycomb, and this type of, it's named colors, a type of regions of the chromatin that likes to be together. When you put that, you recover quite a lot of the high-C contact, not all of them. But in the simulation that I showed you, high-C are put again. So it's once we have the micro-C, we do the high-C to actually cherry picking again all the things that were compatible with the micro-C data. And at the end of the day, we don't have a simple, we have many potential solutions of the chromatin fold. But yes, I mean, high-C is not predicted at that time, is introduced. So
I suggest we continue discussion over a coffee break. So thanks again, Modesto. There have been